Everybody. Welcome back to this series of videos where I go through the progress checks for the AP units that are going to be on the exam. So right now we are hip deep in Unit 4, which is cell communication and cell cycle. This is the second FRQ question from the progress check. And it looks to me like this is about the cell cycle. So chapter 12 in our textbook. This was the very first thing that we did this semester. So right at the beginning of our Unit 3, genetics. So scientists use cells called fibroblasts to study factors that cause non-dividing cells in the G0 stage of the cell cycle to re-enter the cell cycle and undergo mitosis. In a laboratory, fibroblasts typically grow and divide when they are cultured in a growth medium that contains 10% serum, the liquid part of blood that remains after blood clots. If fibroblasts are cultured for 48 hours in the absence of serum starvation, they stop dividing and enter the G0 stage of the cell cycle. The scientists divided serum-starved fibroblasts into groups to which they added no serum, 10% serum, or 0.1 serum. Each of the groups with 0.1% serum was also treated to block the production of only one or a combination of four proteins, P16, P18, P21, and P27, that are inhibitors of cyclin-dependent kinases. The scientists grew the fibroblasts for 48 hours and then measured the amount of DNA replication taking place to determine whether the cells had re-entered the cell cycle. The scientists compared each group of cells to the group cultured with 10% serum and plotted the data. And that's what you see here in figure 1. So the important thing to know here is that G0 is going to be the stage of the cell cycle where everything's kind of on hold. And we kind of talked about it. It was there was the circle diagram of the cell cycle, and then G0 was sort of off up on the northeast corner where it was just like nothing's happening. DNA is not replicating, cells not growing. It's just in stasis. It's doing its job, it's metabolizing, it's producing things that it needs to produce, but it's just not replicating its DNA. It's not preparing to divide in any way. It's on hold. So let's take a look at what they want us to do with this. Okay, and then as you recall, just to kind of give you a framework for this question, when we looked at the control of cell cycle, there are those checkpoints that are controlled by cyclin and these cyclin-dependent kinases, right? And then, once you have enough of these proteins, right, what we will have is the cell going through the checkpoint and going back into the next part of the cell cycle. Okay, so based on the data for the groups of cells cultured with 0.1% serum, so groups 3 to 7 over here, identify which of the four proteins tested do not appear to play a role in blocking cell cycle entry by fibroblasts. Okay, so really all we have to do is we have to remember that these are inhibitors, right? So if they are present, they should be lowering. They should be lowering the amount of DNA replication that's happening. So it looks to me like the only one with significant drops in levels, like these might a little bit, right? But it looks like the one with the significant drop is going to be this group 3, and the only inhibitor that is not present is going to be P16. So it looks like if you take P16 away, this is still inhibited. So you could definitely make the argument that P16 is the one that does when it's not present. Replication is still inhibited. Because again, you have to remember that these are proteins that will inhibit the activity of CDK, cyclin-dependent kinase. Okay, great. So let's take a look at B. Based on the data for the groups of cells cultured with 0.1 serum, so again, groups 3 to 7, identify the groups most likely to include cells undergoing mitosis. Well, it's kind of the opposite of part A, right? Because if they're doing mitosis, they're going to be in the cell cycle. So it looks like there's going to be mitosis in groups 4 through 7. And I would say that group 7 is the most likely. Group 7 because there is the most DNA replication going on. And again, even though DNA replication is not part of mitosis, it's going to be in that S part of the cell cycle. You need to do that first before you're ready for mitosis. So there's the most replication there. Okay. Let's see what they were looking for. So, okay, group 3, in which P16 is blocked, does not appear to play a role in blocking cell cycle entry. And then most likely be, okay, so 7, or in groups 4, 5, 6, and 7. So either way, you could either go with the highest one or those that are all significantly higher. Again, without error bars, it's a little hard to tell. But since they don't give you those, either one would be an accurate prediction. Okay. So here we go. So neurons, nerve cells of the central nervous system, brain, and spinal cord are in G0 of the cell cycle and generally do not divide in response to an injury. So the scientists predict that they can use the data from this experiment to help them treat individuals who suffer from spinal cord injuries. Provide reasoning to support the scientists' prediction. Well, so if you look, what they've done is they've blocked the inhibitors of the cell cycle. And based on their data, it looks like they did so successfully. In the fact that when you block these proteins here, right, P18, P21, P27, when you block those, you get significantly more DNA replication, which then would lead to, theoretically, cell division. So you could just kind of say something like blocking the inhibitors. Blocking the inhibitors allows cells to leave the G0 stage, and if that worked, if that worked for these fibroblasts, theoretically, it should work for nerve cells as well. Okay. Okay. All right. So last thing, interactions between cyclins and cyclin-dependent kinases control the cell cycle, as you should be well aware at this point. Explain how the presence or absence of inhibitors of cyclin-dependent kinases might play a role in normal cells becoming cancer cells. Well, this is just like a process that we've talked about before. What you have is, so remember that the cancer cells are cells that divide uncontrollably. So let's just work backwards. So cancer cells divide uncontrollably, and then so they would have to lack inhibitors, right? They would have to lack inhibitors to allow them to go. So you would have absence of inhibitors, which then would lead to no control over cell cycle, or less control over cell cycle, right? Which would then lead to uncontrollable cell division. You guys answered a FRQ very much like this earlier this semester on one of my quizzes. Uncontrollable cell division. Okay. So let's double check here. Blocking some of the cyclin-dependent kinase inhibitors in this experiment made fibroblast divide. Scientists might also successfully block cyclin-dependent kinase inhibitors in neurons to make the neurons re-enter the cell cycle and divide. And the lack of appropriate cell cyclin-dependent kinase inhibitors, lack of inhibitors, might let cyclins and cyclin-dependent kinases continue to form active complexes, sure, that will result in almost continuous cell cycling with DNA replication and cell division. So, yep. So it looks like that's that. Okay, thanks a lot, everybody. I will continue with the Unit 5 project progress checks shortly. Thanks a lot. Bye.